Night closes over the ocean, and a blue spark answers another beneath the waves. Far below sunlight, animals flash, glow, blink, and release luminous clouds. The deep ocean appears to speak in light, but language is a dangerous metaphor. Some signals demonstrably attract prey, deter predators, find mates, or conceal a silhouette. Other flashes remain unexplained, because observers cannot know what the receiving animal perceives. The mystery is how chemistry became information in a realm almost entirely dark. Each message begins when a living system converts chemical energy into visible photons. Natural selection then shapes brightness, color, timing, position, and response around ecological consequences. If you descend through twilight, sunlight fades while biological lights become increasingly important. Blue wavelengths travel farthest through clear seawater, biasing many signals toward blue-green colors. Eyes adapt to those wavelengths, creating receivers tuned to the most common emissions. Predators exploit that sensitivity, prey manipulate it, and potential mates recognize patterned displays. The result is not one universal code, but overlapping channels used by different species. To decode them, evidence must link a visible event to the action that follows. The first spark, therefore, opens an investigation into senders, receivers, deception, and survival. Bioluminescence differs from reflected color because the organism or its symbionts generate the light. Common reaction joins a light-emitting molecule called luciferin with an enzyme called luciferase. Some systems use photoproteins, and detailed chemistry varies among unrelated lineages. Evolution has produced luminescence many times rather than inheriting one mechanism from a single luminous ancestor. Bacteria, dinoflagellates, jellyfish, crustaceans, squid, and fishes all include luminous representatives. Some animals manufacture required compounds, while others obtain them through diet or microbial partnerships. Specialized organs can contain reflectors, lenses, filters, shutters, and densely supplied light-producing cells. Those structures control where photons travel and which observer is most likely to receive them. In upper twilight water, an animal's dark body can remain visible against dim downwelling light. Photophores on its underside emit a matching glow that reduces the silhouette. When ambient brightness changes, output adjusts, preserving camouflage across depth and time. This Q-action adaptation sequence is called counterillumination, a concealment signal rather than a conversation. The receiver is a predator below, and the useful consequence is failing to detect prey. Some squid release luminous material when attacked, producing bright distractions in surrounding water. The flash draws attention away, the squid jets elsewhere, and the predator pursues a false target. Other animals illuminate an attacker, potentially making that predator visible to larger hunters. This burglar alarm hypothesis has behavioral support in some cases, but not every defensive flash. Alarm labels should follow experimental outcomes, not dramatic appearance alone. Luminous prey lures reverse the direction of deception by attracting another animal closer. Anglerfishes carry a modified fin ray, ending in a lure that can contain luminous bacteria. Movement of the lure mimics edible activity. A nearby predator approaches, and jaws close suddenly. The apparent message promises food, while its actual consequence is capture. Light becomes language-like only because another nervous system has evolved to respond predictably. Below the sunlit layers, most bioluminescent emissions cluster in blue-green wavelengths. Seawater absorbs red light rapidly, and many deep animals have limited sensitivity to red. Most signals, therefore, maximize transmission through the prevailing optical environment. Some dragonfishes produce red bioluminescence and possess visual pigments capable of detecting longer wavelengths. This combination may illuminate prey without alerting animals insensitive to red light. The private channel interpretation is strongly plausible, though perception must be tested species by species. Many luminous fishes also carry rows of photophores arranged in species-specific patterns. Potential mates may use these patterns for recognition where body outlines disappear. Direct courtship at great depth is rarely observed, so function often remains an informed interpretation. Sex differences in light organs strengthen mating hypotheses without proving the complete signaling sequence. When individuals approach, timing and movement may carry as much information as anatomy. Flash duration can separate a brief defensive startle from a sustained searchlight. Pulse intervals can distinguish a repeated display from random disturbance. Position determines whether light meets prey ahead, predators below, or partners beside the body. Context transforms the same blue photon into camouflage, attraction, warning, or confusion. Researchers decode context through deep-sea video, spectral measurements, controlled behavior, and anatomical study. Video preserves sequence, but vehicle lights and noise can alter the event being recorded. Spectral instruments measure wavelength and intensity, yet capture may change an animal's natural state. Behavioral experiments test responses, though simplified tanks cannot reproduce the open deep ocean. Anatomy reveals optical design, but structure alone cannot establish the intended receiver. These limitations make spectacular footage the beginning of interpretation, never final proof. A flash near a predator may be defensive, accidental, or triggered by contact. A rhythmic display near another individual may concern mating, aggression, spacing, or something unmeasured. Scientists strengthen conclusions by predicting different consequences and testing which pattern repeatedly occurs. If startled animals flash before predators withdraw, deterrence gains support. If predators redirect attacks toward released luminous clouds, decoy function gains support. Ventral lights track overhead brightness, counterillumination gains support. If you hover in darkness, even a reliable signal faces physical and biological noise. Suspended particles scatter photons, distance weakens them, and other flashes compete for attention. Sensitive eyes solve scarcity, but risk saturation when sudden light strikes nearby. Some animals use shielding pigments or mobile shutters to control exposure. When a bright threat erupts, rapid neural responses trigger escape before detailed recognition is possible. Speed becomes the adaptation, and survival becomes the consequence of interpreting minimal information. Predators can exploit those reflexes by generating flashes that provoke revealing movement. Prey can exploit predator attention by coordinating light with rapid changes in direction. Evolution, therefore, creates an arms race between detection, concealment, mimicry, and resistance. The deep scattering layers contain immense numbers of small fishes and crustaceans migrating each day. Their movements transport carbon, while their luminous interactions shape who eats whom during the journey. At dusk, migrants rise toward surface food beneath weakening sunlight. Counterillumination adjusts during ascent, reducing exposure while feeding opportunities increase. Before dawn, descent restores dark refuge and carries consumed carbon into deeper water. Bioluminescence thus affects ecological connections, extending far beyond an isolated flash. Dinoflagellates near the surface emit light when water motion deforms their cells. A passing animal triggers a glow, revealing its outline and possibly attracting a larger predator. That response can discourage grazers, although outcomes depend on species and ecological setting. Human disturbance also triggers displays, creating glowing wakes that are beautiful but biologically costly. For the organism, every photon consumes chemical resources and may reveal location to unintended viewers. Future evidence must record natural exchanges without flooding the scene with artificial illumination. Low-light cameras can capture timing while minimizing disturbance, though sensitivity still imposes limits. Spectral measurements should accompany behavior, so perceived color is not guessed from ordinary video. Experiments can vary one cue at a time, then compare responses across predators, prey, and mates. Anatomical findings should generate predictions about beam direction and receiver position. Repeated field sequences must confirm that laboratory responses occur in moving, three-dimensional water. If you return to the opening spark, its meaning now depends on everything around it. A glow-matching downwelling light erases a body rather than announcing one. A dangling point promises prey, while concealing the predator attached behind it. A luminous cloud redirects pursuit, buying seconds for a soft-bodied animal to escape. A red beam may expose creatures unable to see the hunter, illuminating them. Other flashes still resist explanation, and responsible interpretation leaves them unnamed. No single dictionary can translate deep light because each lineage evolved within different relationships. Yet natural selection repeatedly converted photons into cues that change behavior and survival. The ocean's luminous language is real as information, while its many untranslated signals remain scientifically open.